Okay. What am I doing? What have I got? Oh. Crap. Haemostasis, like homeostasis, keeping your blood level at the same level. Okay, that's what stasis means, I think. But you know the gist. So we're going to be talking about clotting factors, the clotting cascade, and generally what a normal blood vessel looks like. Okay. So I'll start off with what a normal blood vessel looks like. So we've got our three main layers. We've got our tunica intima. So that's our innermost layer of our blood vessel. Okay, that's the closest part to the lumen. So the lumen is obviously the middle bit where the blood flows through, the plasma. After the tunica intima, we have the tunica media. Okay, makes sense. It's in the middle, tunica media. And then the third layer is the tunica adventitia. Not sure why it's called that, but it just is. Okay, so in our tunica intima, we've got three subsections. Okay, so this will make sense. Well, it's easy to look on this big vessel I've drawn out here. We've got our endothelium, this red layer, which shows the endothelial cells, and that's the bit that makes direct contact with your lumen. We've then got our subendothelium. So this is mainly collagen, quite high collagen levels in your subendothelium. And then you've got your internal elastic lamina. Not really sure what that is. I think it's like a basement membrane. But just know that that's the third bit. Okay. Imagine it's quite elastic, hence the name. Okay, in our tunica media then, it's mainly smooth muscle. Okay. So smooth muscle in this area, and that can contract and relax for vasodilation, vasoconstriction. We've also got, in between the tunica media and the tunica intima, we've got these little green things that I've done here, and the orange up here that you might not be able to see. And those are your nociceptors. So these are our pain receptors. I don't really know much about pain at this point in time, but they're important in this context because when we have a cut or something broken, when our blood vessel is broken, that's going to trigger our pain receptors, our nociceptors, to contract our smooth muscle. Okay, that's as far as I'm going into it today. I don't really know much else about pain and how it works. Okay. And then we've got our tunica adventitia. So that's connective tissue. And its main purpose is to sort of support the vessel and connect it to its surrounding tissue. So that's the bit that sort of blends into our outer border. Okay, so that's our main sort of structure of a blood vessel. Okay, and obviously in arteries compared to veins, we'll have different proportions. So don't quote me on this, but I think in an artery, the lumen is narrower, so thicker smooth muscle. But in the vein, you've got a wider lumen, smaller muscle, I think. But might have to double check that. That's just off the back of my memory. Okay. All right. So I'm going to briefly look at the classical model of our clotting cascade. Come over to this side. Okay. So the classical model is what happens. Well, it happens in the body, but this is when they discovered it. They were doing it outside of the body. So this is the process they found when they were doing it outside the body, but it slightly varies in the body, and it's more natural in the body, if you know what I mean. So things happen and different interactions are happening at the same time. So it's not as clear cut as this classical model makes it seem. So the cell-based approach is the generally accepted one, but we still have to know the classical model because that's still relevant. It's just not exactly what happens in the cell. So overall, this is what happens. But this doesn't include our cell interactions in the body. Okay, so we've got our intrinsic pathways and our extrinsic pathways. Intrinsic pathways, that happens if our blood vessel's damaged, okay? So if we've got a damaged blood vessel, that means, actually, I'm going to come back to this. I'm going to talk about what's happening in a normal blood vessel without damage, and then we'll go back to that, just because it'll flow a bit better. Okay, so we've got our normal blood vessel. We've got our tunica intima, and our tunica media I've put on here. I haven't bothered with the other one. But we've got three things that keep our blood from not clotting. Otherwise we'd have clots all the time. Okay. So firstly, our blood's flowing this way. So it's going that way through the bloodstream, and all these things are floating in it going through the blood, in just your everyday blood vessel. So our endothelium, this secretes two things, and that is PGI2 and nitric oxide. Okay, so that's important because it inactivates our platelets. So if the platelets are inactivated, that means they're not going to be clotting or attaching to the walls of our arteries, okay, or veins. So that's important. First step is inactivating the platelets. So no platelet plug can form. Okay. We've then got these receptors on our endothelium layer. These purple receptors I've got here, and they're heparin sulfate receptors. Okay. So what they do is antithrombin 3 attaches to it. Okay, attaches to these receptors, and it sort of acts like a massive knife and cuts all these clotting factors as they pass through it. So it inactivates clotting factor 2, 9 and 10. Okay, so let's look where are 2, 9 and 10 in this classical model, which we'll go on to in a minute. So 9 is here, 10 is there, so 10 is quite an important one, it's here to the middle, and 2 is here as well, so 2 is also a very important one. Okay, so in inactivating them, so this process can't happen. Our intrinsic pathway and our common pathway can't happen. We've also got receptors here as well on our endothelium, and these are thrombomodulin receptors as well. Okay, so these thrombomodulin receptors, they bind to thrombin. So thrombin is just the technical word for factor 2. So again, factor 2, 2A, sorry. But essentially the factor 2 is going to bind to this thrombomodulin, and it's going to have like this little hand is going to come out, and it's going to activate these proteins. So it's going to activate protein C, which I've drawn as a C there. Okay, so when it activates this C, so I've got a coloured in C there instead. So this is coloured in C is active. When this is activated, this activated protein C inactivates cofactors 5 and 8. Okay, so it stops them from working. Now, where's cofactor 5? Cofactor 5 is here. It's needed for cofactor 10, and cofactor 8 is here, and it's needed for cofactor 9A. So you can see there that these things in a normal blood vessel without damage, these processes are happening to prevent a clot, because we don't want that to happen. Okay. So when we've got damage to our blood vessel, it's going to go through 5 steps. So first we have vascular spasm. So that's basically just contraction of our vessels to try and stop as much blood going through as possible so it doesn't leak out. Then have platelet plug formation. So that's essentially lots of platelets going into the hole to stop blood from leaving. We'll then have coagulation, which is when we've got all these clotting factors, and that sort of solidifies the clot. And then we've got clot retraction and repair. So that's sort of the final phase where our clot is starting to repair itself and sort of our blood vessels returning back to normal. So these endothelium are going to contract a bit more and come back together and repair themselves, so they're going to seal off again. And then we've got fibrinolysis, where the fibrin, which is produced, and that's the bit that makes our clot strong, that's when our fibrin is destroyed and removed. So the clot dies, basically, because we don't want a clot floating away in our blood and going into our lungs or our heart. Okay, so that's what's happening in our normal blood vessel. Okay, so we'll go back to this classical model then. So if in our intrinsic pathway, if we've got a damaged blood vessel, so if it's damaged, so say we've got our blood vessel here and suddenly it's damaged, okay, we've now got endothelia, so all these blood vessels, these red blood cells, all this blood is now able to escape out through here, okay, so our blood is coming out through here. Okay, that's bad. We don't want that. Okay, so what happens is our damaged blood vessels, our endothelia, so this red bit, our collagen and our platelets, they're all interacting. Okay, so our collagen, if we get rid of this arrow now, we know the blood's coming out. We've got our collagen, it's now trickling in here, okay? So our collagen is now interacting with the blood. Okay, so this triggers our, essentially our factor 12, okay? So factor 12 now, it bumps into this collagen and it activates and becomes factor 12A. So when it's got an A next to it, that means it's active. So factor 12A has been made after it's bumped into the collagen. Okay, that triggers our factor 11 to turn into 11A. So 11A is active. And that in turn activates 9. So our factor 9 goes from 9 to 9A. Okay, that makes sense. It's a little chain reaction we've got there. At this point, 3 things need to happen. So our factor 9A, so our activated 9, it needs vitamin K, calcium, and factor 8. Okay. So why does it need vitamin K? Vitamin K, if we look here, this is our polypeptide. So all the factors, they're proteins, and these proteins are made in the liver. So they're polypeptides. And when vitamin K binds to it, it adds CO2. Okay. So we've got this COO minus group. So that makes it negatively charged. So an important thing to note now is our collagen is negatively charged. Okay? So that's important. We've got negatively charged collagen. But if we're thinking, well, we've made this negatively charged now, isn't that not going to repel it? Which is why we need our calcium. So our calcium, it comes along and it binds to this negative charge. So if we think we've now got our COO minus binds to our calcium, this calcium is now able to bind to something else. That something else is our collagen. Okay. All right. It then also needs, so we've got our vitamin K and our calcium. It also needs cofactor 8. So cofactor 8, it binds to our 9A to make a complex, and that's cofactor 9A/8. Okay. And this converges on cofactor 10. Okay, so this point where they converge is X or 10. X marks the spot. That's where they converge. And they like Roman numerals because why would they make it easy? Okay. Let's look at them. We'll look at the common pathway in a minute. Let's look at. Actually, no, we'll look at the common pathway now. So our common pathway then, that's triggered. We've got this 9A/8 complex, and that triggers our factor 10 to activate. So it now becomes 10A. Okay. Our 10A now needs 3 things. This needs vitamin K, calcium, and factor 5. So, as discussed before, we've got our calcium and our vitamin K, which allows it to bind. Because why do we want it to bind? We want it to bind so that it doesn't drift off. If we've got our factors coming in, they're doing all this while they're moving, and suddenly we want them to be sucked in to this negatively charged collagen. We don't want them to go all the way down here and start clotting. We need them to be attracted there. So suddenly we've got loads of factors. We've got factor 9A, that's going to be on our collagen. We're going to have factor 10A, that's going to be on our collagen. And they're all just going to be waiting to react with other molecules or factors that are coming along. If they're stationary, they're more likely to be hit or collided with. Okay, so that's why we need our vitamin K and our calcium. Factor 10A also needs factor 5, so that forms a complex again, and once this has happened, it can now trigger factor 2, which is prothrombin, to turn into thrombin, or factor 2A. Okay, so when we've got factor 2A, again we need that vitamin K, our calcium, to enable it to bind to our collagen. That initiates our fibrinogen, or factor 1, to turn into fibrin. Okay, so why do we need fibrin? Fibrin is important as this is the sort of like stuff that builds our clot. Okay, so this is in the coagulation phase. I need to point this out now. Okay, this is after our platelet plug. This is coagulation, but I'm just running through our classical model first before we go into the cell-mediated sort of response there. Okay, so our fibrinogen turns into fibrin. This is really important to keep our clot in shape and keep it with a nice structure so it doesn't break off and float away. Okay, and fibrin, it interacts with factor 13 to form loads of cross links. So if we think we've got this polypeptide, this fibrin, which is a polypeptide because it's a protein, it's now going to form loads of cross bridges, and so it's now branched, which makes it more useful when it's holding together the platelets, the platelet plug, which is why it's important. Okay, so let's look back then and look. We've seen how the intrinsic pathway works and how it gets to fibrin being made. Let's look at our extrinsic pathway. So that happens when we've got tissue damage. Okay, well you might think, well, how, if we've got a damage to our blood vessel like I've just shown here, surely we're going to have extrinsic interaction? Well, yeah, you might have a bit of both. But generally, if you've got sort of, you can have blood vessel damage without them being ruptured and that sort of thing, which is why you've got two types of clots, or two pathways. So we've got our extrinsic pathway, that's due to tissue damage. So what happens is, in our tissue, we've got tissue factor, which is essentially a factor 3. So this is floating around, factor 3, and it enters, use the wrong colour there, that's not really easy to see, but it enters into and interacts with our blood. When it comes into contact with our blood, we've got factor 7. So factor 7 is in our blood, and this comes along and interacts with factor 3. So if we've got factor 7 interacting with factor 3, okay, that means our 7 is going to turn into 7A, so it's going to become activated. Again, we need vitamin K and calcium for it to bind to our collagen, and then this can interact with our factor 10. So here we have it again, binding to our common pathway, and that's where they all come together. So they converge there. And again, we've got the same pathway from here. Our factor 10 becomes factor 10A. That needs vitamin K and calcium. And factor 5, that allows prothrombin to turn into thrombin. So factor 2 to go to 2A, which also needs vitamin K and calcium, which allows it to stick to the collagen. And that turns our fibrinogen into fibrin. So anything with ogen is going to be inactive. Okay, that's in our inactive form. And then we've got our factor 13 making our cross links. Okay, so generally that's what's happening in our coagulation with the classical model. Good way we can remember this is we've got in the middle we've got our 10. So if we look over here, we've got our 10. Factor 10 is in the middle. Factor 10 is in the middle. In our intrinsic pathway, we're going to be going sequentially down. So if we start with 12, we're going to go 12, 11, we skip 10 because that's in the middle, 9, 8, and then 10, okay? So that's sequential on this side for the intrinsic pathway. For the extrinsic pathway, we need factor 7 and then 3. So if we add them together, we get 10. And then from our common pathway then, if we've got 5, then 2, then 1. So if we times them together, so if we multiply them together, we should get 10 as well. And then final step, we've just got factor 13, okay? So just need to remember that as sort of an outlier, and it sort of happens at the end to make those cross links. Okay, I think that's it for our healthy vessels and our classical model. Okay, I'll do another one which outlines what happens in our vascular spasm and generally our cell-based approach for a clot.